Okay, guys. So I just had a question about the recitation quiz. So everyone knows there's a quiz after every recitation, right? Okay, because you all read your syllabus, right? So I had a question about that. So the quiz is based off of the paper that you read and the discussion questions that you go through. The quiz is at the end of recitation. So don't worry if you're reading the paper and you have got no idea what it's about, because hopefully during recitation, the questions that you discuss within your group, because it's going to be small groups working together, working through a list of anywhere from 5 to 10 questions. And some kind of activity related, be it some practice problems or some kind of drawing activity or something. And so ideally, at the end of those 2 hours, you do understand the paper, you do understand everything, so that when you take the quiz, you'll be like, oh, no problem. Okay? So the quiz is really a motivator for you to A, prepare, read the paper, and B, participate and go through these discussion questions. Okay? Any questions about that? Bring your Scantrons, because you can't take the quiz if you don't have your Scantrons. Okay. So, business, right. Recitation starts on Monday. So I've already posted the first paper on Blackboard, and there will be discussion questions posted by sometime on Sunday for that paper. Don't worry about that. You don't have to go through them in advance. You just need to bring the paper with you. Or if you have your laptop, you can bring your laptop and have that with you. That's fine too. But you just want the questions, because if you don't have the questions, you can't discuss anything, then you can't prepare for the quiz, and then bad. Bring your Scantrons. The other thing, added students. There's about 9 of you who are added to the class. I think everyone is now on Blackboard. Is anyone not on Blackboard yet? Okay. So I will open pre-test and quiz 1, and you'll have till Wednesday to do them. The rest of you, if anyone was confused or whatever about any of those, you can still go back and look at them if you want. You can't take them again because it's the first grade that's recorded, but feel free to look at them. And I will try to fix the ability to see the correct answer on quiz 1 so you guys can get that. What else? Quiz 2 this weekend. Okay, so the guys, the people who just added, you have 3 quizzes to do, but you have more time. So, okay. So let's go over quiz 1. We're not going to do all of the quiz, but what I do is I look through the answers and see how you guys did, and questions that like 15, 20 of you or more got wrong, I'll go over in class. So here's one. One gene can result in many different proteins with many different functions. This is mostly due to, actually, before we do this, I want everyone to take a piece of paper, or 4, and write A, B, C, or D on it. And you guys are going to hold up your answer, because what I find is, this way I could really see what people think, whereas when one person goes A, then I don't really know what the rest of you guys are thinking. And I already know how you answered on the quiz. So already I know a lot of you didn't get this right. And I was like, I should bring, what are you guys doing? Wow, very good. Okay. I was thinking I should bring Sharpies, and no. Just use a pen. I could see that. That's good. A, B, C, D. If you want, you can do an E too, because sometimes there's an E. And then if you want, you could do an all. Okay. So one gene can result in many different proteins with many different functions. This is mostly due to, I'm seeing lots of A's and C's and D's. So that means there's a lot of confusion, which is why I put this up there. So the answer is all, right? So one gene, so that's a sequence of DNA, with a start and a stop of some sort. one gene can result in many different proteins. So RNA. So then you need to start thinking to yourself, okay, what needs to happen for a gene to become a protein? So you need transcription, translation. What's happening at the RNA level? What's happening at the protein level? And which of these things make sense, right? So alternative splicing. So those who put up A, that is correct, but there's more than just that, right? Alternative splicing definitely results in different proteins. RNA editing, which we'll learn about today. Adding U's, deaminating or inserting U's that will change either reading frame or generate early stop codons. Post-translational modifications, which we'll learn a little bit more about today, that will post-translational, so this is a modification on the protein, right? So that will determine whether the protein is going to be active, whether it's going to be stable, where it's going to localize to. So those are all ways that you end up with many different proteins with different functions, okay? And so we'll talk more about that today, and hopefully any questions that remain will be understood. So I'm partially going through these to help you guys start to think about how to tackle the types of questions that I ask. So hopefully this will help. Okay, mRNA lacking a 5' cap is likely to be found in this part of the cell. This was slightly upsetting that not everyone got right. Lots of C's. Lots of C's. So I guess all the people who are here today, 1 D, 2 D's, 3 D's. Okay, C's and D's. Okay, so you guys are smart because you don't think cell wall, right? Because that was like a throw it right out. And plasma membrane, right? So those were both like dead giveaways. But C and D, it seems like there's some confusion. So let's remember that RNA, when you have RNA, it needs to be processed and that become a mature RNA, which is capping and polyadenylation and splicing, and in order to leave the nucleus, right? So if you're lacking your 5' cap, are you going to be able to leave the nucleus? No. So that's how you would think through that. Okay. So some of you guys might be sitting there going, but you didn't even teach us this yet. But so if you're looking at this and you're like, 5' cap, what is that? Google it. Look it up in your textbook. Whatever you need to do to find out what a 5' cap is and what it has to do with export from the nucleus. I'm trying to teach you guys how to research things and teach things to yourself because you're going to remember it. You know, like when your mom's like, go look it up. You always remember the answer, right? Or your dad might say that, or a friend, your teacher. Anyways, okay. Small RNAs such as siRNA and mi microRNAs have the following function. So a lot of you guys probably never really learned about functions of RNA other than becoming mRNA and protein, but this is a big one, and if you didn't know it, you can easily Google siRNA or miRNA. B's, D's. Okay, so activation of translation, definitely not. So A and D are no. And, A and C are like the same thing. That was really bad on my part. So B, mRNA degradation. So that's a big thing that happens. And a major way of controlling whether or not a gene is going to actually become a protein, be expressed and become a functional protein. And we'll learn more about that today. Okay, so far. This is what we talked about last week and the other day. So what needs to happen or not happen to get a functional protein, right? That's what we've been going through, and that's what we're going to finish up today, right? So you need transcription. You need transcription to actually happen, right? If there's no transcription, you'll definitely not get a functional protein. And there's a whole bunch of regulation about transcription, which you learn about if you take my 409. RNA maturation, capping, splicing, polyadenylation. It needs to get out of the nucleus. It needs to bind to the ribosome, right? Will an mRNA that doesn't bind to the ribosome ever become a protein? No. Okay, just making sure everyone's paying attention. Then the other thing is RNA stability. If your RNA is degraded, Will it ever be translated? No, right? So a lot of this is very logical when you start to break it down into these kind of simple ideas. So why are there many more proteins than genes? So we've been talking about that, right? One gene, many proteins, many functions. So that's what we're talking about. Why can one protein have many different functions? And that has to do with post-translational modifications, and we'll learn more about that today. So I try to do this in the beginning of each lecture to kind of reorient you to what we were talking about. And if you always go back to this slide before every exam, go to each lecture, pull up this slide, this will give you categories. These are main big picture topics that I tend to think about when writing questions. I go back to the quizzes, I go back to these kind of summary slides. Bless you. And so when you're asking me for a study guide or you're filling out your evaluation at the end of the semester and you're like, I'm upset that she didn't give me a study guide, I want you to remember that you have a study guide by putting together all of these slides and by looking at the quizzes, right? Okay. So alternative splicing, we spoke about that briefly, but this is where we're going to continue. So a lot of proteins can be produced from one RNA. If you leave certain introns in, obviously you're going to contain more codons, right? You're gonna acquire more codons. So there's so an example is in Drosophila. There's one example. Now it's an example and it's an extreme example, so this is not normal. But there's actually 38,000 proteins that come from one transcript. When I say transcript, I mean mRNA. 38,000 different proteins from one mRNA. That says a lot about the power of alternative splicing, right? So that's from different things. One is alternative splicing, and then also RNA editing, which we'll talk about in a minute. So you can have different kinds of alternative splicing, right? One is where you might have an optional exon. So if you think, so these little V things means that's what's spliced out. So the intron is the yellow. So an optional exon might appear, right? So normally, like maybe in some cases, these splice sites are recognized and this whole exon is not there or is there in the alternate mature mRNAs, right? You can have an optional intron, right? So maybe sometimes this intron is spliced out and sometimes it's not. Sometimes it becomes part of an exon. Are you okay over there? I know that's not, the placement of this is not ideal. Sometimes you can have mutually exclusive exons, right? So sometimes you might splice here, so you'll remove this exon, and then sometimes you might splice here, and you'll remove this exon, so completely different transcripts again. And sometimes you'll have an internal splice site that's not always recognized, and so sometimes this intron will be spliced out, and sometimes this larger intron will be spliced out. So it's just all different ways it can happen, but you can see how you can have a huge amount of variation of proteins that come from this, right? Okay, so different cell types will do things differently. So the other thing is, so don't, like some of you guys can be really good at listening and writing, but if you're feeling challenged about listening and writing at the same time, stop writing, because what's the point? It's all recorded, right? It's all online. So, you know, I always would get really confused if I tried to listen and write. I can't speak and write either. Okay. So another way, right? So another way of having many proteins from one RNA is alterations in transcript cleavage and poly A addition, right? So if you, and we're going to have a nice example here. So at the end of your mRNA, at the 3' end of your mRNA, that is actually formed when you have cleavage of your transcript. So you've got RNA polymerase doing its thing, and then an enzyme comes along and cleaves it off. Okay, and that's what gives you your 3' end of your mRNA. And depending on where that enzyme recognizes and where it cleaves, you can have an mRNA of different lengths, right? So an example, so this actually is done on purpose to give you different proteins sometimes. So an example is in B cells. Who knows what B cells are? or I should say who doesn't know what B cells are? Okay, good. So B cells, antibody secreting cells, immune response. So this has to do with antibodies. So in a naive B cells, a B cell that has never seen any sort of pathogen or antigen of any sort before, they have membrane-bound antibodies. So it's actually a membrane protein that's an antibody. And then after they see an antigen, B cells mature and they secrete antibody. And that's very efficient, right? So it's kind of like they're selected based on their ability to bind whatever invading pathogen is there or allergen. But then and then they're kind of selected, they mature, and then they secrete tons and tons of it. Very efficient, right? So this is regulated by increased expression of a cleavage factor. So as a B cell matures, this cleavage factor is the expression of it is also increased, and it recognizes a poor cleavage site. And it cleaves the sequence that encodes the transmembrane domain. So I'll show you what this looks like. I think this is very cool. So IgM, that's the first antibody usually that's made. So here's how it looks. Here's your DNA, and you've got an exon, a poly A site, another exon, another poly A site. And so when you have the membrane-bound form of IgM, you end up with this RNA. So 3, 4, 5, 6. Obviously, there's 1 or 2, but we're not paying attention to it because we care about the end of it. And then when you have mature, when you mature, you express more of this cleavage factor, and it's going to suddenly recognize a different cleavage site, and you only get exons 3 and 4. And so that results into you get splicing and maturation, and you end up with the longer form of the RNA in the naive B cell, and that results in this membrane-bound IgM, and that's actually cleaved off in your mature B cell, and you end up with, so it cleaves off the transmembrane anchor, and it's actually secreted mRNA. Yes, mRNA, IgM.
What do you mean by membrane-bound antibody versus what?
Secreted.
Okay.
Yep. Yep. So here's your membrane bound. So it actually has a transmembrane domain, a really hydrophobic domain that's going to be embedded in the plasma membrane. Yes, definitely stop me if I speak about something that's like really confusing or I'm vague. But if you cleave off your transmembrane domain, are you going to be in a membrane? No, right? Because there's no hydrophobic domain of the protein to be embedded in the membrane. So then you're secreted. And we'll learn more about how this all happens shortly. Okay, so this is all based on recognition of a cleavage site. And the cleavage factor, that expression of that cleavage factor is increased when a B cell becomes a mature B cell. Any questions about that? Yes?
We're talking about actually cleaving the antibodies from the membrane?
No, we're talking about mRNA. Thank you for that question. So she asked, are we talking about cleaving the antibodies from the membrane? No, we're talking about mRNA. Okay, so at the RNA level, so you get transcription, and then usually there's a cleavage factor that actually cleaves the RNA, and then it comes off of, like, RNA polymerase keeps going, but there's a cleavage factor that cleaves it, so the mRNA is released, and that is spliced and polyadenylated, and all that needs to happen, and then exported out of the nucleus. So there could be 2 cleavage sites that are recognized. One is recognized in a naive B cell, and that allows for the transmembrane anchor to be there, and the other one is recognized in the mature B cell, and it's recognized because the cleavage factor, so the enzyme that's doing the cleaving, is upregulated, so is produced at higher levels in mature B cells. And so suddenly this, at the mRNA level, it's being cleaved, and so there are no codons for the transmembrane domain, and so you get a protein that completely lacks the transmembrane domain. And if there's no transmembrane domain, you can't be embedded in the membrane. And then, as we'll see soon, membrane proteins and secreted proteins are in the same pathway, and so it gets secreted out of the cell. Yes?
So the mRNA that's being made gets cut from the RNA polymerase before it can transcribe the transmembrane part of the.
Yes.
Okay. So And the RNA polymerase keeps going?
Yes,
Does
and
that mean that the transmembrane part of the mRNA is still transcribed but like separately floating around somewhere?
it might be. Yeah, so the question was, so if the cleavage site is before the transmembrane domain, then does RNA polymerase keep going, and is there some RNA for the transmembrane domain floating around somewhere? Possibly, I'm not 100% sure, and chances are if it is, it's strange and doesn't look right and is degraded in the nucleus. Any other questions about this? Does everyone understand? I'm using this as an example. So I use a lot of examples so you can apply a concept. And so this is an example of alternative cleavage and how it results in two different proteins. Okay, because antibodies are proteins, right? Questions? Thoughts? Opinions? Okay. Everyone loves it. So that's just another way of looking at it. I'm not going to go through it, but I put it here because sometimes one figure doesn't do it for me, and I need to look at another figure to think about it. So there you go. This actually maybe makes it more clear that we're talking about, I like the other one because it showed a picture of the cell and the antibodies, but here it's a lot nice. It's nicer if you need to understand RNA, then translation happens, and then you have protein, right? Okay. Okay, so another thing. So remember, we're talking about how different genes can result in many different proteins. So another mechanism for that is RNA editing. So for example, insertion of U-nucleotides in RNA transcripts. So it can be insertion of U-nucleotides, but it also can be deamination of cytidines. So an example of that is ApoBec1, which is a protein. It's a cytidine deaminase. So can you guys tell me what the function of a cytidine deaminase is? You all should know, otherwise you need to leave. It's in the name. Deaminates cytidine, right? ACE enzyme. So it deaminates cytidine, right? What's cytidine? Okay. Are you guys being shy? I love to feel stupid, okay? It's so fun because it's liberating, right? I feel stupid. It's awesome. So, yesterday I was asking for something and I was like, that silver thing that pipettes go in? And they're like, you mean the can? And I'm like, can. Not silver thing. So we're all like dumb sometimes and it's hard to put yourself out there. But do it because it's liberating because you'll be like, you know what? That person was a total idiot yesterday. I can be an idiot today, right? I'm not going to judge anyone. Right now I'm secretly judging all of you because you're scaredy cats. No, I'm just kidding. Well, kind of. Okay, cytidine deaminase, right? Deaminase. What did deaminase do? Think about it. It's in the word. Huh? I heard. Remove nitrogens. Okay, it's kind of right here. Yep. Yep. So, amine group. Who's taken chemistry? All of you. Who's taken orgo? I know, I'm sorry. My condolences. I hated orgo. Who's taken biochem? Someone? Okay. It helps. I never asked, how many people are cell molecular people? Okay, how many people are OBE? I've had OBE students kick butt in this class. I just want you guys to know. So, I'm looking at both of you. How many people are the other one, the A and P? Okay. How many people are not bio majors? You're not? Oh yeah, MB3. Right. Yeah, that's bio. I know, it's different. Okay, anyways, just making sure I'm not like talking to someone who's like a sociology major and has no idea. Okay. Cytidine. Okay, again, cytidine. What's cytidine, guys? Is it amino acid? Okay. So see, we have, those nucleotides that make up the DNA. Nucleotides make up DNA and RNA. Yeah. So cytidine. Okay. So Cytidine deaminase, ApoBec1, that's the function, right, of ApoBec1. It deaminates cytidines, so, and makes them into uridine, sorry. So here, right here, is cytidine, and there's cytidine deaminase, and it removes this NH2, and so C to U, right? That's what it does. So in ApoBec1, with ApoBec1, it actually mutates the C in the CAA codon, which is glutamine, to U to make a stop codon. Okay? What would making a stop codon do? Stop. Stop. Translation. What if you make a stop codon early in a protein, what would it do? Make it longer or shorter? Thank you. Okay. Oh God, you people. Wake up. Okay. So here's how it works. You've got your RNA, and then ApoBec1 comes along, and it edits it, and it changes this glutamine to a stop codon, and you end up with two forms of a protein, right? Because it's not 100% of the time that this editing happens. It happens in certain cases, right? Because genes are only expressed under certain conditions, and so ApoBec1 transcription and translation will increase. You'll have more of it around under certain conditions, like maybe when you need to make LDL, because actually it edits a gene called ApoB, which is involved in making LDL. You know what LDL is? Who's had their cholesterol tested? Low density lipoprotein, right? So ApoBec, when it edits it, then, when it doesn't edit it, it makes LDL, and when it does edit it, it makes a shorter version, which is chylomicron, which is involved in something else, I forget. But one gene, one RNA, edited two proteins, okay? Fun? Are we having fun? God, it's so hard in the beginning of the semester for me because I get these blank looks all the time, and it kind of scares me. Okay. So we just talked about a whole bunch of stuff. Okay, so export of RNA from the nucleus. mRNA needs to get out of the nucleus to make the protein. If it never gets out of the nucleus, it never makes a protein, right? Only 1 in 20 RNAs are exported from the nucleus. So if you've got introns, or you have no cap, no poly A tail, you're retained in the nucleus, and you're degraded, right? Okay, so here's an example of how a virus, has anyone heard of the HIV virus? It's a really obscure virus, and it only infects tortoises. Okay. HIV, so viruses are really good at getting around the cell's protective mechanisms. Not just immune response, many things, right? So here's a story about HIV. So it encodes a protein that allows for export of intron-containing transcripts. Now you might say, okay, so what? What does that mean? Well, think about it. So you've got a virus, and it does its thing, and it gets into the nucleus. The genome gets into the nucleus. You'll have transcription. Transcripts come out of the nucleus, just like with our own transcripts. You get translation and proteins, right? But don't forget, what happens in the nucleus? The replication of the viral genome, right? So HIV is an RNA virus. So you have multiple RNAs that are being made. You have these spliced RNAs that are being shipped out and made into proteins. And then you've got unspliced RNAs that also make different proteins because viruses are really efficient. And then you also have whole genomes that need to get out of the nucleus and packaged and made into virus, right? Because the whole point is to make more virus and get out of the cell and go infect another cell, right? So is everyone understanding the problem here? We have whole genomes containing what? Introns, and lacking what? Caps and poly A tails and all sorts of things. They should not be able to get out of the nucleus, right? They should be stuck in the nucleus. So HIV was really smart, or, well, smart for the virus, but unfortunate for us. And And so HIV encodes a protein called REV. I'm not going to test you on this. It's more so you can understand things. It binds to an RNA sequence, so a sequence within the genome called REV responsive element. Who cares? It's a little hairpin, creates a nice binding site for a protein, and it interacts with a nuclear export receptor, and we're going to learn all about nuclear import and export later, and kind of fools the nuclear export machinery to exporting out of the nucleus. So why do I tell you about this? Because there are ways around everything, and viruses are real. I love viruses. Viruses are really good at getting past our quality control systems. Okay. Questions? Yes. Why doesn't it get degraded when it's out of the nucleus? Because it doesn't have poly A and all that stuff. Well, okay, so you can't get out of the nucleus when you have introns and you're lacking poly A tail and 5' cap. But once you're out of the nucleus, well, what's going to happen? Well, you're not going to associate with the ribosome. You're not going to be translated. So most RNAs that hang around too long and nothing happen, well, they do get degraded. But the whole goal in the virus is for it to be packaged in a new virus and go out of the cell. So it goes and it interacts with another protein and then gets packaged into a virus and just goes. So it's not hanging around for a very long time. Maybe some might get degraded. But it's being packaged and and basically leaving the cell. It might be. So it interacts with GAG, I believe. Who knows this? Who's taken virology? I think it interacts with a viral protein GAG, and I believe that takes it right to the plasma membrane where it starts to get ready and assemble to be transported out. Okay, good question though. Okay, so different fates. What time is it? Good. Different fates of RNA, right? Okay, so RNA, it's now out of the cell. I always want to call the nucleus the cell for some reason. It's out of the nucleus. Okay, so now what? It gets bound by the ribosome and translated, right, in the cytosol. RNAs that contain certain signal sequences, those are directed to the ER for the secretory pathway, and we're going to learn about that in great detail shortly. Other RNAs will associate with cytoskeletal proteins that take them to different locations, because don't forget, it's really ideal for RNAs to be shipped to where they need to be functioning when they become proteins. So they'll be shipped to different places and translated and imported into mitochondria or nucleus or wherever. And that is determined by sequence between the stop codon and the poly A tail, which is called the 3' untranslated region. So I'll say UTR. UTR means untranslated region. There's 5' UTR and a 3' UTR. So that means there's actually the stop codon, and then there's a bunch of sequence, and then you have a poly A tail. So somewhere within that region, there'll be a sequence that will help in targeting where things are going to go. Okay, so this is just what I said. RNA is localized to parts of the cell where the resulting protein will be used. So here's just an experiment. Let me turn this off. Where people were studying what part of the RNA is responsible for localization. So what they did is they basically chopped off the 3' UTR. They probably chopped off a whole lot of things when they were doing the true experiment, but what they put in the textbook, I think this is straight out of the textbook, they only show the pretty stuff, right? They only show you the data that tells you something. But so if you look, so they've labeled the RNA with something fluorescent, okay? The red RNA contains the 3' UTR, and the green RNAs, the 3' UTR is deleted, so they chopped it right off. And so you can see right here, red is localizing right here. This, I believe, is in Drosophila. So it's localizing to this part of the cell. And where is the green localizing to? Nowhere. Whenever you have diffuse staining, it means it's kind of just everywhere. It hasn't been targeted anywhere. So you're going to learn how to look at these, and it'll start to make more and more sense. So you can see the red was targeted to these blue circles, which is some part of some like developing Drosophila embryo or something like that. But the 3' UTR deleted here, and it's completely diffused. So that tells you that the 3' UTR is required for targeting of this RNA, at least, to where it needs to go. Okay? I wish I didn't have to turn lights on and off. I need like a remote thing. Okay. So if you were studying RNA, so I want you guys to take 1 minute. Does everyone know the person you're sitting next to? No? Okay, stop, take a minute, introduce yourself to your neighbor. And I want you guys, everyone kind of sits in the same place all the time. So you guys will end up being close to each other frequently. So introduce yourselves, and frequently I'll ask you to discuss something with your neighbor. So I want you to think about how if you were studying some RNA, and you needed to you wanted to know whether or not this RNA is required to go to the nucleus or, I don't know, somewhere strange in order to function. How would you inhibit RNA localization? So stop, chat. Hi, my name is Bob. My name is Jane. I like whatever. And then discuss the question. Yes. You three of you can work together.
But I guess you can like start going into getting rid of destroying other
I mean, if you could just even have with prime UTR, do that.
Okay, guys. Did everyone meet your neighbor? I saw some people not talking to anyone. I understand some people just don't like to talk to people, but it's good to know people because sometimes that person might become a good person to study with. I highly encourage studying together. Do not study by yourself because that's like studying in a vacuum, and if you'll just propagate bad information. It also is nice to have someone to take quizzes with, even if you do it like over Skype. Google Hangout, anyone? Okay, so how would you inhibit proper mRNA localization? And who wants to tell me what they think? It's a really easy one because I kind of went over it.
Cut out the 3' UTR.
Right, so cut off, yeah, so the thing is, so this is assuming that sequence is in the 3' UTR, but yeah, chop up the RNA, right, and determine what doesn't go to the right place, yeah.
Couldn't you also somehow like forcefully edit the UTR cap?
Oh, yeah, so what if you had an editing enzyme of some sort, some kind of, I don't know, like maybe deaminase, because maybe it's a string of sequence and it's likely to have some C's, maybe. So some kind of editing. Yep. Okay. Anyone else? Okay, so this kind of, I like you to apply information to kind of experimental questions. So it's good to start thinking that way. Okay. Proteins can bind to the 5' or 3' UTR, right? Remember, both ends, to negatively regulate. When I say negative regulation, I usually mean inhibit, right? Inhibit translation. So that makes a lot of sense if you think about it, right? Because your ribosome needs to bind to your RNA. If you've got a big protein blocking it, that might inhibit the ribosome from assembling on the RNA. If the ribosome can't assemble properly, you're not going to get translation, right? So that's another mechanism that's frequently used to regulate translation. Okay. More things for translation control. Phosphorylation of the translation initiation factor, EIF2. We're not going through what all the different factors are, but there's the translation initiation factor, which is required for assembly of a ribosome, and if it's phosphorylated, then you can't assemble your ribosome. So that's one way of controlling whether or not translation is going to happen. Remember, we're talking about what you need to do to get a functional protein. You can have multiple AUGs, right? If you have multiple AUGs, AUG is the start codon. You get multiple start sites, you're going to have multiple proteins, right? Because the start sites might be in different frames, right? So that's complete different proteins. Or they might just, like, you might have shorter or longer proteins. Usually it's different frames. And then internal ribosomal entry sites. So that would be some kind of start site that's like smack in the middle of the RNA. Okay. Okay, so then another thing that could regulate or control whether or not you're going to get a functional protein is mRNA degradation, right? So you can control the outcome with either stabilizing or degrading your RNA. So most RNAs have a short half-life. Who's ever worked with RNA? Who's ever heard nightmares about working with RNA? Yeah, some people have. Okay, so RNA is degraded so easily because RNases are everywhere. And so people always are like, oh my God, be very careful when you work with RNA. But actually, it's more stable than you think. But anyways, but they do have a short half-life. That should be less than. Anyways, so there's two degradation pathways that I'm going to talk about in the cell. One is poly-A tail shortening. So your poly-A tail, not only do you need your poly-A tail to get out of the nucleus and associate with the ribosome, well, not associate really with the ribosome, but anyways, but one thing that can really shorten the half-life of an RNA is getting rid of that poly-A tail. And that's sometimes done purposely. If you want to tightly regulate the expression of a certain gene, you're going to keep that poly-A tail short so that that RNA doesn't hang around too long. Because if the RNA goes away quickly, then you're not going to be able to make a lot of protein, right? Is everyone following that line of reasoning? The other thing you can do is sometimes you can add more A's to your poly-A tail, so that will increase the half-life of your RNA. Now, you're not going to do it. It's not like you're like, I will add A's to my poly-A tails. No, I mean, it's in the cell, right? Right. Okay, everyone got that? And then there are specific endonucleases that just chop off poly-A tails. And it's all regulated. It's not like there's this endonuclease going around chopping all poly-A tails. This is very gene-specific, transcript-specific, because you're going to regulate not only whether or not your RNA is made, but how stable it is and whether it's ever going to be translated. Okay? Then there's nonsense-mediated decay, and that just senses RNAs that somehow made it out. And there's stop codons that shouldn't be there. It's more of a quality control, and it degrades RNA. So then we'll talk about these small non-coding RNAs that affect mRNA stability. So these are the siRNA, mi, microRNA, piRNA. Bless you. There's a lot of sneezing. I think it's ragweed season. I've been feeling it. Just so you know, ragweed, taking Allegra. Okay, so they affect translation either through direct inhibition or through, so directly inhibiting translation or through destabilizing, targeting mRNA for degradation. So that's frequently done viruses like to do that. Viruses encode a lot of microRNAs that will specifically degrade cellular RNAs. It's a really nice way to shut things off that you don't want to be on, right? If you're a virus, what don't you want on? What would you like to shut off if you were a virus trying to successfully infect a cell? Immune, anything immune, right? Apoptosis, maybe, right? Because you want the cell to survive long enough to complete your life cycle, right? Those are common targets. Okay, so this is how these small RNAs work. I'm mostly going to focus here on microRNAs and siRNAs. This is the whole pathway, but don't worry too much about that. What I want you to think about here is, okay, so you've got your transcription and you've got these microRNAs that are made. They're these kind of hairpin structures, and they come out of the cell. Oops. They come out of the cell, associate with proteins. Don't worry about what, but they cleave, they trim, they help in maturation of these small RNAs. And what you have in the end are these single-stranded, short, like 21-ish base pair, single-stranded RNAs that base pair with mRNA. So it's sequence specific, right? And they'll base pair with whatever mRNA they're targeting and target it for cleavage. So then the mRNA will be degraded and it won't be able to ever be translated. Another way things work also is by binding. So you'll again have this single-stranded RNAs that bind to the mRNA and prevent association with I believe prevent translation. I forget exactly how, or it might not completely be known, but prevent translation, and then ultimately RNA is degraded. So one's cleavage and one's translational repression. But both very, very common ways. It's like there's an explosion of microRNA research. So it's very important for you guys to appreciate that they exist and that they are important in regulating gene expression, right? Because if you're degrading RNA, then you're not going to get protein. Any questions about microRNAs? Anyone wondering why I'm talking about them? Okay, 2 more minutes. Protein activity control. Okay, post-translational modifications control protein activity. They affect protein stability, right? There'll be protein or it'll be degraded. If it's degraded, it can't function, right? Localization, if proteins don't go where they need to go, then they can't function. And interactions with other biomolecules. That's a nice big list of different kinds of post-translational modifications. You need to memorize them, and I'm going to quiz you right now. Are you done? Do you not memorize this? Just appreciate it. I'm a big fan of ubiquitination, so make sure you understand what that is. So the other day, I had a question about multiple post-translational modifications on one protein, right? Well, here's the example that I was talking about with P53. So P53 is a tumor suppressor, right? And as you can see here, let me turn off the light, and then I'll let you go away. As you can see here, each P53 protein, these red ones, you see there's insane amounts of post-translational modification. Here it's phosphorylated, and it's phosphorylated, it goes here and associates with this, and it goes here and associates with this, and it goes here and associates with this. It's acetylated. When it's acetylated, it goes here and it's associated with this. It's sumolated. It's ubiquitinated. It's, what else is it? I'm sure there's more. Acetylation, nitrosylated? No, netylated. This is netylated. So as you can see, P53 does a ton of things, right? There's your nucleus, there's your cell. It's involved with apoptosis. It's involved with DNA repair. It's involved with cell cycle. It's involved with transcriptional regulation. It's involved with, it's involved with a lot. And that is one protein completely regulated by its post-translational modifications. What I mean by post-translational modification is covalent modification, so addition of another protein covalently to a protein. Okay, covalent. So it's not gonna fall off. Okay, you guys can go.